Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between molecular therapy methods and clinical developments Editor-in-Chief, Dr. Mohamed Abou-el-Enein, and Drs. Daniel Stone, Harrison Dulin, and Keith R. Jerome from the Fred Hutchinson Cancer Center. They will discuss an article recently published in MTMCD by Stone and colleagues titled Serum Factors Create Species-Specific Barriers to Hepatic Gene Transfer by Lipid Nanoparticles in Liver Humanized Mice. This article will appear in MTMCD's upcoming special issue, Lipid Nanoparticles for Cell and Gene Therapy, which will feature a wide array of original research articles and reviews covering the development and therapeutic applications of lipid nanoparticles. If you enjoy this episode, you might be interested in our hybrid event this fall that will allow you to engage with pioneering researchers in gene editing. Breakthroughs in targeted in vivo gene editing will be held in San Diego and virtually November 20th to 21st. Visit asgct.org/breakthroughs for more information, and we hope to see you there. And now, Drs. Stone, Dulin, and Jerome.
Hi everyone, and welcome to our podcast. I'm excited to be joined today by the authors of a new study just published in our journal. Their new study addresses actually one of the field's most persistent challenge, which is the limited ability of animal models to predict human outcomes, particularly when it comes to gene delivery. As we know, lipid nanoparticles are a leading platform for delivering nucleic acid therapies, for example, but their performance doesn't always translate across species. This study uncovers why they fail in liver humanized mice and finds ways to overcome those barriers. Keith, Dan, Harrison, thanks so much for joining us today.
Thanks for having us.
Yeah, good to be here.
Pleasure.
Dan, let's start with the origin story. How did this study come about? What problem were you trying to solve?
So in Keith's lab, in our group, we are studying persistent viral infections, such as hepatitis B, herpes simplex virus, HIV, and we've looked into other viruses as well. And we, as part of our hepatitis B work, had set up in-house a liver humanized mouse model, which we published some work in with the aim of developing curative therapies for hepatitis B. Now, Keith has set up a collaboration for some of our other work with a company called Excision Biosciences, who we partnered with on this study, and Ryo Takeuchi, who's a co-first author on this study. Basically, he used to work at Fred Hutch, actually, in the lab of Barry Stoddard, so we knew Ryo, and Keith and Ryo basically were talking about maybe doing a study in our mouse model using CRISPR-derived therapeutics to target hepatitis B as a therapeutic. That's what Excision works on. They're using CRISPR stuff to target viral infections. So they approached us and said, hey, we'd be interested in testing our therapeutics in your model. We're interested in using LNPs. And there are a couple of different models which you can use for this. We're using the NSG-PIZ liver humanized mouse model. There are other models such as the FRG mouse model. And they asked us if we just do like a pilot study just to see, our LNP is going to work in this model before we did any, like a long-term study. So that's kind of how it initiated.
Thank you, Dan. This is very interesting. And I think for our listeners, it would be very, very nice to explain what exactly is a liver humanized mouse and why it is such a valuable model.
Yeah, no, that's a really interesting question. So these kind of models have been around for probably about 20 years, give or take, and they're used for a number of different things. Some of the first studies they were kind of developed to study from more of a toxicology perspective. So obviously, when you give a drug to a human, a novel drug, you want to see if there's any side effects, and the liver is one of the main places where you see these effects. So they were kind of initially developed for toxicology stuff, but then people realized that certain diseases could not be modeled in, a mouse or, another experimental lab species. So diseases such as, hepatic diseases such as viral infections, hepatitis B, hepatitis C, malaria, they all contain phases of, their replication and infectious cycle that require a stage in a human hepatocyte, and it can't be done in a dish. It has to be done in an animal. So basically, what you do is you transplant a mouse that has an immune deficient background. It normally has a genetic background as well that is detrimental to the mouse hepatocyte selectively. So the mouse hepatocytes are sick and dying off, and then you transplant them with human hepatocytes, which then basically take over the liver. So you get this chimeric liver that has some mouse and some human hepatocytes in there. Different models use different kind of selections to do this. In the NSG-PIZ model that we use, it's caused by a deficiency in, it's an introduction of a human gene. It's a deficient form of the human alpha-1 antitrypsin gene that actually causes the hepatocytes to be sick, the mouse hepatocytes. So that's how the selective pressure is gained here. So we were one of the first people to show, in this particular model, we were the first people to show that you could use it to study hepatitis B infection a couple of years ago. People have done this in other models such as the FRG mouse, the UPA skid mouse. So this is not a new thing per se, but this particular model. And what we like about this model is you can get the mice from Jackson Labs. They're relatively inexpensive, and you can do this in-house relatively cheaply. You can generate these mice. If you buy them from the companies, they cost thousands of dollars per mouse. So, it's just a much easier way for an academic lab to go about something like this. And we set this up in-house, and it works really nicely. And yeah.
Thanks, Dan. And talking about hepatitis B infections, my question is actually to Keith. So, Keith, your lab has long worked on chronic viral infections. Why is hepatitis B still such a high priority for curative therapies?
Yeah, well, hepatitis B is actually a really interesting target. As you know, every virus has a different biology. So the question about what you can actually plausibly aspire to cure really has to have some serious thought put into it. So, we know there's been a big effort in HIV cure, for example. There's some real challenges there because the reservoir is everywhere in the body, and it's big and it's hidden. We really like, for example, herpes simplex as a target just because there's so few latent viruses and they're in neurons in a very distinctive localized place. So hepatitis B is somewhere in between. Not latency, but the persistence is actually limited to the liver. So there's one target you need to get to. And in general, gene therapies being delivered to the liver is something that we as a field have worked on, right? So things like to go to liver, right? Even if you don't want them to go to liver, they go there. So there's some real pluses. And then finally, look, we work at the Fred Hutch Cancer Center, and there are hundreds of millions of people with persistent hepatitis B infection, and they're at markedly increased risk of hepatocellular carcinoma. So it's just something that is biologically plausible, and it's a really clinically important virus. So we're really excited to have these models to work on it.
Thank you so much, Keith. I hear you, and I think delivery, since it's a central point in your story, I'm really excited also to maybe shift to Dan and ask, you did use LNPs as a choice for the liver gene delivery. Can you explain more why and why not use AAVs, since also you and Keith are known to work on AAVs and have a lot of experience with?
No, that's a really great question. And, we've been thinking about this for probably about 10 years. AAV is great for the liver, has so many pluses, but it actually, in the context of a treatment like this, it has some negatives, which, we've thought about, but, reviewers of grants have definitely reminded us about this. The nature of having, long-term expression of some form of nuclease such as CRISPR that is basically continual expression, you have this ongoing risk that there might be, some off-target related activities due to, indirect cleavage of, similar target sites within the human genome. So there are concerns in relation to that. But also, there are potential concerns in relation to, can something like that also increase the integration frequency of hepatitis B, which is really one of the main drivers of hepatocellular carcinoma and hepatitis B. So really, it's like the duration of expression is an issue here. Now, you could look at that as being a plus, right? The longer you have expression, the more likely you are to cleave all your viral DNA and get rid of your target. But it's kind of it's a double-edged sword. So what we looked at as an alternative to this to LNPs is we thought, if we can go in with a short, LNPs, you get expression for a few days and then the mRNA goes away. So just having like a short burst or several short bursts would minimize some of the safety factors there. So really that was kind of our rationale for looking at this as well. We haven't given up on AAV. We're still we're doing some head-to-head studies currently, but moving forward from a safety perspective, I think LNPs would be probably favorable in a lot of regards.
Thanks, Dan. I agree. I think LNPs have great potential in so many applications, and their safety for sure profile has been shown to be favorable. So when you tested different LNPs in your mouse models, what did you see happen when the mice got the LNPs?
So why don't I cover this little first bit, and then I'll kind of Harrison can take off from here. So initially, we working with Excision had partnered with Acuitas, who very well known LNP manufacturer. They're involved in one of the COVID vaccines, and we were sent 3 proprietary LNPs that just that expressed with a GFP mRNA for the initial analysis. We don't know the chemistry behind those ones, other than that we know they're ionizable lipids. And our initial studies showed that basically, they went very nicely to the mouse hepatocytes, but we didn't see any transduction to the human hepatocytes, which for our work with hepatitis B is essential, right? If it's not in the cells with the viruses, you're not going to get a therapeutic effect. So that was kind of our initial results, and then we kind of went into started looking into finding out why. And then, from there, how we could kind of get around that. And really, that's kind of where Harrison joined us at this part of the project and has really kind of taken this on. I also want to mention Michelle Lopriano, who also did, is a co-first author on this work with us. And she did a lot of this initial work with me and then the later work with Harrison.
That's great. I think, Harrison, let me bring you in here. And as a postdoc driving a lot of the in vitro work as well firsthand, maybe you can explain more as when did it become clear to you that LNPs weren't reaching the human hepatocytes, and what kind of experiments helped you to figure this out?
Yeah, so Dan and Michelle and the other members of the lab had been doing this work with the Excision LNPs, and that was right about when I joined the lab, was when they were getting some initial data that showed the human cells weren't getting transfected. So I got there and I was like, this is interesting. I want to know more about why this is happening. So I actually got a hold of a separate LNP from GenScript that we could get a lot more of it to do more experiments with, that was based off of the SM102 LNP. And we put that in a mouse again, and we saw the same thing as the other LNPs that we tested. It was not going to the human cells in vivo. And so from there, I was like, okay, this seems to be not just specific to one hepatocyte donor or one particular mouse model or one particular LNP formulation. This is very broad. And let me just take this one SM102 LNP and from there kind of see where we can go with it. And the field had known for a while that serum factors for conventional, 4-component LNPs require serum factors in order to target liver. The most prominent and well-studied of these factors is ApoE and other lipid binding proteins, these direct LMPs to liver hepatocytes. So we knew that serum factors could play a role in that. There had also been some very cool studies showing serum factors being able to redirect targeting of lipid nanoparticles to different organs and different cell types. So I was like, okay, serum is easy to get. And so let's go ahead and get some and see from the mice and see if that plays a role in how the hepatocytes are getting transfected with our LMPs. So yeah, I went in vitro. We took the SM-102 LMP and plated some primary human hepatocytes, the same ones that go in the mice. And we saw that the serum from the chimeric mice dramatically inhibited transfection of primary human hepatocytes in vitro. And not just with the SM-102 LMP, we got a hold of a couple of other LMPs as well, using a few different formulations. All of them were dramatically inhibited by the chimeric mouse sera. So it was like one of these things where you see like very clear expression and then just no expression. It was very your listeners can look at the figures. It was pretty obvious that there was no transfection in vitro. So yeah, that was an exciting day in the lab for sure.
Oh, that's great. Harrison, so thank you. So once you saw that the serum factors could block the delivery of the LNP, so you started looking closely at how human hepatocytes were or were not getting transfected. What did that reveal about how LNPs interact with their environment? And did this shape your strategy in a way?
Yeah, so again, yeah, lots of people have been working on how LMPs can target specific cell types. And so we knew that serum factors were especially important for targeting different cell types. And most of the LMPs that people work with and that are available have four components. So they've got cholesterol and a PEG lipid, a helper lipid, and an ionizable lipid, right? That's like how we traditionally think about what goes into making an LMP. And then those lipids are getting recognized by lipid binding proteins in the sera to get you your targeting to specific cell types. So I was there and I was thinking, okay, so in our chimeric mouse model, that step of binding to the serum factors seems to be messed up, or something is blocking it, something is interfering with it. We actually, from our experiments, we set up a competition assay in vitro that showed it's not just that we're missing something in the sera, that there's actually something present in the mouse sera that's actively inhibiting the LMPs in these chimeric mice, which was surprising. But we were like, okay, that's surprising. We need to get around the serum factors somehow if we want to target our primary human hepatocytes in vivo.
That's very exciting, and I'm really, congrats on this great work. And so let me ask you, did you end up finding a formulation that actually could bypass the block and successfully transfect the human hepatocytes?
Yes, we did, which was a very exciting day in the lab. I just was talking to the lab members about this yesterday, and when I got this data, I wanted to run around the lab shouting Eureka to everybody. But yes, we did. Again, like the idea is that we need to get around whatever is inhibiting the LMP in the sera in order to target the human cells. So let's make the logic went. Let's get an LMP that can directly target the human hepatocytes without having to rely on binding to these lipid binding proteins in the sera. So we got a hold of an LMP containing decorated with tri-GalNAc. So GalNAc as a moiety on the surface of the LMP can directly bind to the surface of the human hepatocytes without having to rely on any factors in the sera. So we put this tri-GalNAc LMP in the mice. Lo and behold, we got transfection of the human hepatocytes in vivo. It was a good day.
Congrats again. This is very exciting work. And maybe I can ask you, Keith, what does this mean for others using the liver humanized mice to study, human targeted gene delivery?
Yeah, I think the big take-home message here is don't just assume that everything's going to work in these models exactly like you might predict, or that you've seen in another model system, or that you've seen in this particular cell type in a different setting. Like you truly do need to validate these models extremely well. And I think we sort of point a pathway forward that should people see these sorts of things with their particular LMP. And again, as Harrison said, this seems to be a pretty broad observation. So people really need to look for it. But I think that should people run into similar problems of the lack of transduction of their human cells in vivo, that there really is a really nice candidate approach. And again, with this trigalNAc direct targeting, I think Harrison showed us really very strong transduction. It's actually equivalent between the cells, and it's really, really got around this problem. So not only a problem to look for, but I think a really nice solution. So we're really excited because now we get to move forward and actually think about using these models for what we wanted to do, which was to try to develop curative therapies for hepatitis B.
Thank you, Keith. And I know that your lab has been leading efforts on curative strategies for chronic infections for years, and maybe you can tell us more about, outside of this project, what else are you excited about right now, and what are you working on in your lab?
Yeah, well, we're pushing kind of the lead viruses, as I mentioned, are this hepatitis B work and also the herpes simplex work. So we're really excited about moving those forward and getting to the point, we can start to think about clinical translation, which is very exciting, and get some of these into humans. One thing we've been pushing hard on is really understanding the spectrum of genomic effects when animals, or ultimately humans, are exposed to a gene editing approach. So really, what are we doing? What are the off-target effects that we see, and how do you minimize that? So we have projects, for example, that have been really pushing hard on tissue-specific expression of nucleases, so we can really limit their expression only to the places that need to work, and have demonstrated in other studies that almost completely eliminates off-target effect. For example, in the herpes work, we can really now just limit the gene editing to the neurons, and we see essentially nothing happening off target in the liver, which is a place we'd always worry about. And so we can do those sorts of things for hepatitis B as well. That's really exciting. And then a little more forward looking, we're still really excited to use some of these gene editing approaches, but really as targeting for the viral genomes, and then sort of take away the gene editing aspect and think about epigenetic modulation of these genomes, this idea of deep latency. That's been explored some for HIV, but relatively less for hepatitis, and almost none for herpes simplex. So really excited about that we may have, first of all, just a couple of shots on goal, kind of complementary approaches, but also have an approach in our bag of options that doesn't require double-strand DNA breaks. I personally don't think those are going to be a showstopper for gene editing in vivo, but we always have to keep that in mind. So we've got a couple of different approaches, and I think they're all super exciting. And then the last thing I just have to throw out there is some work that Marius Walter's been doing in the lab with some gene drive work for herpes simplex, and we'd love to think about how to do that for hepatitis B, but it's such a tight genome. I don't know if we can do that or not, but we certainly have some fun discussions. But that's really excited to basically use versions of these viruses themselves with their native tropism and all the things they've learned to do, and now take that and use that to actually fight against the disease that these viruses cause.
Thanks, Keith. I think this is really a very compelling direction, and maybe just as a closing point to our listeners, what do you see as really the biggest barrier right now to turning those mechanistic insights into durable clinical responses? Is it, you talked about off-target effects, we talked about delivery. So is it more about the delivery, persistence, or something else entirely, or a combination of many?
Yeah, I've always thought that the big barrier here is effective delivery. I mean, to me, that is problem number one. And that's why we've loved, as you pointed out, the work with AAV, because it's really good at this. And again, they're very tissue specific, and you really need to look for your application and try to understand what's the most important thing. But I'm also thinking kind of a step ahead. And should we have success and find therapies that can actually cure these diseases, particularly in people, what are they going to cost? Can we actually get them to everybody who needs that? As you know, hepatitis B, for example, is a problem in really resource-limited parts of the world often. And if we come up with a therapy that has tens or hundreds of $1,000s in manufacturing costs, that's going to be hard to roll out and hard to get to people who need it. So one of the reasons I've been excited about lipid nanoparticles is that in theory, they might be more scalable, they might be more economically reachable for folks. And so I think we'd be remiss in not evaluating those early on and be thinking along these lines.
Perfect. This is really a very important aspect for the entire gene and cell therapy field, and how we can really ensure delivery, not just actual delivery in tissues, but also delivery to other parts of the world and patients in general. Keith, thanks so much, and thank you to all of you, Dan and Harrison. This has been really an insightful conversation, and your work really shows how, preclinical models can mislead us if we don't understand the biology behind them and how we can fix that. Thank you again for joining us. Congratulations on your publications. And to our listeners, you can read this article now in MTMCD. And thank you for listening, and see you in the next episode.
Thanks for having us.
Take care.